Next week, Jonathan will come in again and teach embryology and heredity. So that's our last piece of material next week. Embryology and heredity is fun. We're really closing up this term with topics that have to do clearly with reproduction, but now they're going to move into development. So reproduction, we know a decent amount about because you can dissect people, you can look into their organs, you can figure out through a microscope, oh, is this a gland? Does this look like just some kind of bladder that just holds things? So you can really get an idea of kind of the inner workings of the reproductive system. What's really hard to do is figure out what happens during development. That's what we're going to do next week. And development is everything from the fertilization, sperm comes in, fertilizes the egg, and everything from that point on is considered development. So when that part is really, really not well understood, it's a really interesting thing to look at because it's kind of like a story more so. It's not just, it's more like next week you're going to be really learning kind of the story of development. And there's a really cool video, some of you maybe have already seen this, it's called Life's Greatest Miracles. It's in Miracle. It's narrated by John Lithgow. It's really nice. And it's a really amazing cinematography that is put together separate still images and a couple of video images of actual developmental processes going on inside of a person. It was done in the late 80s. It's never been beaten since. It's just one of the hardest things to actually get a camera on and actually view. So it's a really cool thing. You'll have an option to watch it at the end of next week's lab. You can also just find it on YouTube and watch it. Kind of have a movie night this week. Again, it's called Life's Greatest Miracle. And it's really fun. It's a really good thing. And it also uses the same terminology we use in this class. So the terms that you're going to learn next week will just kind of be reinforced by the Life's Greatest Miracle video. And I know it's on YouTube. I watched it a couple of years ago on YouTube. So unless it got taken down, I don't think it did though. Other than that, next week you will not be having a quiz. Next week you will basically be doing embryology, heredity, and you'll be graded on attendance. So you do need to be here. You'll get 10 points for attendance, whether you're here. Well, if you're here. If you're not here, you'll get 0. So make sure you're here, because that's pretty nice to get 100% on that. And then the next week we're going to be doing the practical, so that's when you'll see me again. The practical will begin probably at 12:30, so you can show up early for your practical and get it out of the way early. And then you can show up as late as 1:25 p.m. Try not to be any later than that. So practical should be really, really similar to everything we've been doing up until this point. It should be pretty evenly distributed in terms of how many questions for each of the 4 weeks. But urinalysis was kind of a short week. So there'll probably be slightly fewer total urinalysis questions. They're going to be just like your quiz. I mean, you're going to be reading a urinalysis output, and that's it. But there'll probably be fewer, probably more like 10 of those questions rather than 13, which would evenly distribute it. And those questions that are not on urinalysis will probably be on reproductive anatomy instead because there's just so much material from last week. So you'll probably have a little bit more reproductive anatomy, a little bit less urinalysis, and then probably equal numbers of questions on embryology and heredity, and what we're talking about today, which is reproductive physiology. So make sure you're prepared with reproductive anatomy. Spend some extra time today reviewing that kind of stuff. That's definitely the bulk of the anatomy. The new information today is a lot of histology going through the ovaries and the testes. That's what we're going to be looking at and talking about today. So I think that's it for the practical. So again, same setup. You can show up early, you can show up a little late. And that's it, I think. Anything else? I think that's all I wanted to talk about before we get into today's new material. All right, so first thing we're going to talk about is a little bit of review. The first section of your packet talks about meiosis, mitosis, what it means to be haploid, what it means to be diploid. All of these terms we haven't really talked about in quite a while. Biology 112 introduces these concepts to you. Today we're going to return to it. Most of it's review. There are 4 terms that I want you to not know about, and they're on the next page of your packet. But while they're on my mind right now, go ahead and flip to the second page at the very top. There are 6 terms. The middle 4 I don't want you to know. Dyad, let's see, synapsis, tetrads, crossing over, and dyads. We don't really have a good visual representation of those things, so I don't think they're really worth your time. However, I do want you to know what a stem cell is, and know when DNA replication occurs. And since we're looking at that right now, let me just cover that for you. DNA replication occurs prior to mitosis or meiosis. If you remember that, it actually happens specifically in what's called S phase. But the big takeaway message is to know that it happens prior to mitosis or meiosis. And if you think about it, remember, you got to replicate the DNA before you split the cell in half. Because if you don't do that, one of the cells won't have DNA. You got to copy it first and then distribute your 2 copies of DNA into separate cells. And so that happens before you even go through cell division. You've already copied the DNA. So that's kind of what that main point is just to remind you of that. A stem cell is an undifferentiated cell. And what that means is it's a cell that still could become a different type of tissue. So for instance, early on in embryological development, you've got a bunch of cells clustered together, and they haven't decided yet whether they're going to be muscle cells, nervous tissue cells, epithelial tissue, or connective tissue. They haven't differentiated yet. So a stem cell is really valuable from a medical point of view because it gives you an opportunity to take a cell and turn it into something else. And so it's kind of magical that way. And the reason you can do that is because if you remember, every cell in our body has every gene that it needs to be any kind of tissue it wants to be. And so it doesn't have an inner emotion that make it feel like I want to be epithelium. There's just some signal that happens during development that converts a stem cell, and it says, oh, okay, I'm going to be epithelium now. And then it usually goes down that path and stays that way. It rarely will go become epithelium, be your skin for 10 years, and then turn into another type of tissue. If it does that, that's called metaplasia. It's a very dangerous thing because often it's a sign of impending cancer because it's a cell that's kind of gone rogue. It's not doing what it's programmed to do. So rarely do you see cells ever undifferentiate, so go become something specific, and then go back into being a stem cell. But there's a lot of research looking into how to make that happen. Because right now, if your liver fails, you get a liver transplant, which is an option, but it's a pain because you got to take it from someone else, right? So you got to have to actually have someone die with a healthy liver, and then have an airplane rush to go pick up the liver, get it to you with a team of surgeons, and put it in you within a couple hours. And so it's an amazing thing to see happen, but it's not the greatest option in terms of organ replacement. But stem cells, if you could take, skin cells, turn it back into stem cells, and then somehow program the cell to turn into liver cells, you could potentially heal your bad liver by inserting stem cells into it. And so they've started to do this. Plasma, an example of one kind of stem cell is platelets are actually a stem cell. They actually have an ability to differentiate into different types of collagen and elastin fibers. And so if you've ever heard of a platelet-rich plasma injection, that's one of the ways that they're trying to use it. Jury's still out of whether or not it works that well, but there's a lot of effort to figure this problem out. The second most famous researcher at OHSU specializes in this. 2 years ago, he was able to turn basically placental tissue back into multipotent stem cells, which means cells that could become anything. So huge accomplishments being done around here in this field. But it's a big, big topic, stem cells. So that's what that differentiation term means. You're going to hear that undifferentiated or reverse differentiation as a fancy way to talk about controlling cells and getting them to turn into other things. So with development, you're dealing with undifferentiated cells. So if we come back to this image, and let's review haploid and diploid right now. That's the next term I'm going to go to. It's back a couple terms. If you remember, every normal cell in our body, our normal somatic cells, are all represented as diploid, or with numbers, it's 2N. And that means that the N is how many versions of DNA you have. So in almost all of our cells, all of our normal cells in our body, our non-sex cells, we have 2 complete copies of DNA. You could make 2 totally different people out of us. If you were to split the DNA up and somehow get it to engineer into separate people, it'd be crazy, right? You could take my dad's DNA and my mom's DNA, and every one of my cells has 2 of those versions. It doesn't have a third extra version, and it doesn't have a single version. It always has 2. And that's what diploidy is. We are diploid organisms. We inherit 1 copy of our DNA from dad and 1 copy of our DNA from mom. And again, that's represented as 2N. The reason it comes up this week is we're going to talk about how you take normal cells in your testes or ovaries, and you have it actually go through mitosis first, the normal splitting of cells, and then you send that very same cell through meiosis. And if we remember from Biology 112, the whole goal of meiosis is to produce not diploid cells, but haploid cells. So that's what the single N represents in either the egg or the sperm. Because what's going to happen, the reason you don't want those to be diploid is that the egg and the sperm produced in separate living organisms are going to come together to form a new, unique diploid cell down here that we call a zygote. So after fertilization, after the egg and sperm come together, the process is called fertilization, you form a zygote and you return to a diploid state. So here's you over here. You're going to go through meiosis, produce either a sperm or an egg, and then fertilization will take place and you'll return, and this will be the new living organism over here. And so that's what that ploidy, that ploidy is fairly simple for us because we're either diploid or we're haploid. Some organisms are tetraploid. There are occasionally errors made during meiosis that result in sperm that have either no N, they would be missing a chromosome, they didn't get one, or they got an extra one. So for one of the chromosomes, there was an extra one in there. Those are called aneuploides, which means wrong number in Latin, the wrong number of chromosomes. And so those are things that we deal with in our society. Down syndrome is a very common one because people are, most other chromosomal abnormalities result in death. Down syndrome does not. And so in our society, that's where you begin to see chromosomal abnormalities the most. The most common chromosomal abnormalities happen with sex chromosomes, though. Abnormal numbers of the X or Y chromosome are 10 times more common than Down syndrome. So ideally, you don't have any wrong number. You go from haploid into diploid. And that's the overall concept that should be totally reviewed from 112. But doesn't mean it's fresh in your head. Right? That was a long time ago. If we look at this in a little bit more detail, ignore the words, just look at the image. I want to just, a lot of times people remember mitosis and meiosis and they blur them together. So this image is in here to make sure you guys can really separate what's going on in these two processes. We're going to be talking about meiosis mostly. But as a reminder, mitosis, with a T, mitosis, you're taking the cells, you're copying the DNA before you go into mitosis, and then you're going to split the cell in half and make two absolutely identical cells. You're photocopying cells. That's the whole goal. You want the cell to be the exact same. I scratch my skin right here. Oh, I need to replace some skin from below. I want identical cells just to be replaced right in there. That's what mitosis is going to be for. Meiosis, though, it starts out pretty similar. You go from one cell to two. But if you look at the color patterns in these illustrations, there's an important difference from the outcomes of meiosis 1, the first cell division in meiosis, compared to the outcome of mitosis. With mitosis, both of these cells have two different colors in them. And what that's indicating is that these cells have a mom and dad version and a mom and dad version. That's why they're 2N. But when you come out of meiosis 1, you are one color or the other. You either have dad's DNA or mom's DNA, not both. And so the product of meiosis 1, when you're done with the first cell division, you're actually haploid. And it's not really you anymore, actually. Your body doesn't even recognize those cells as self anymore because these two cells do not look the same in terms of their antigens as this cell did. This is a self cell. These two are non-self cells. So you have to be very careful in your body when you go through meiosis 1 because you're immediately producing cells that are non-self. In the testes, you do this behind a barricade that's formed by a cell called the Sertoli cell, and it protects all the products of meiosis from your own immune system. In females, the secondary, when you become haploid, happens so much later on in the process, it doesn't seem to be as big of an issue. But when we look at the ovary and the testes, you'll see that transition from diploid to haploid is a pretty important one. And then meiosis 2 is really simple. All you're doing is splitting up each one of the chromosomes and putting them in their own cell. So the product of meiosis in total, in males, would be 3 haploid sperm. In females, you have, sorry, did I say 3? I meant 4. I can't count. You have 4. You actually make 4 from 1 in males. In females, you actually don't do this. In females, you have four possibilities of cells that you can create, but from any one original cell, you actually only randomly select one of these to produce. So the probability is the same in males and females. You don't do different like genetics and Punnett squares for males and females for most of the chromosomes. But in reality, these three of these, let's say this was the chosen oocyte, which is the name for an egg in humans. If this was the chosen one, these three would actually still be created, but they'd be really little, and they'd be these cells with only chromosomes in them. And we call them polar bodies. So that's one term on your sheet. And a polar body is kind of a wasted cell, and it only happens in females. And the reason is because females are not only trying to go through meiosis, but they're trying to make one really big oocyte. Males are all about quantity. Females are all about quality in terms of the production of meiosis. So they rob the cytoplasm and all the organelles from these three, and you stick them into your chosen cell. And again, these leftovers are called polar bodies. They still have DNA in them. They're haploid, but they don't have cytoplasm, they're non-functional. And so again, that's what a polar body is. All right, so let's take a look at this in a little bit more context in a process called spermatogenesis. So take a look at that. I need to get some water. My mouth is drying out like crazy. Take a look at this and focus on the transition between meiosis 1 to meiosis 2, and take a look at the haploid 2N into N transition and see where that happens. We're looking at a seminiferous tubule on the right. I'll be right back. All right. So left side picture is just a slightly more detailed view of what we were looking at in the previous picture, just illustrating meiosis. But now all the drawings are actually showing the spermatids, which are kind of like undeveloped sperm, and then spermatozoa here, which are essentially sperm but don't function quite the same way. But over here, what it's trying to illustrate is the seminiferous tubules, which hopefully you got a glimpse at last week. Seminiferous tubules are the majority of the mass of the testes, right? They all drain into the rete testes or rete testes, and then to the epididymis, then to the vas deferens, and then out the ejaculatory duct and the urethra. That whole pathway begins with the seminiferous tubules. And so this is what's actually going on. So this is just a slice out of it. It would really just be a big circle. And in the center of the circle, you would be pumping out sperm, basically. The beginning of the process starts up at the edge, and it proceeds this way because you're actually going down and doing meiosis here. So it begins on the outside with a cell called a spermatogonia, or plural, spermatogonium. Sorry, other way around. Spermatogonia is plural. Spermatogonium is singular. And so you have a little stem cell. This is an example of a stem cell because it hasn't really differentiated into anything yet. So it starts, and it's actually stuck to the edge of the seminiferous tubule. So under a microscope, you'll always find them at the edge. The first thing is a little bit surprising, but don't get too stuck up on it. It has to go through mitosis first. Because if all you did is take this cell, this spermatogonium, and force it through meiosis, there would be no cell left behind to replace it. You would run out of spermatogonia. So first, you copy your spermatogonium, and then one of the results of mitosis, so this is mitosis that you're seeing right here. One of them stays behind, and one of them gets pumped through meiosis to become sperm. Does that make sense? You gotta do that, because otherwise you'd run out of spermatogonia. So along this process, you can see the ploidy, the 2N transitioning to N. It's just meiosis, that's all you're seeing, same thing. Product of meiosis 1, 2 haploid cells. Those 2 haploid cells split again into your 4 sperm right there. So that's nothing new. What's new are the 4 basic types of cells that you see in this process. So there are 4 types of cells I want you to know about. but only really 3 that I'm going to ask you to identify under a microscope. So the first one I already mentioned, that's the spermatogonium. You will find this under a microscope. It's pretty easy. They're around the edge, always on the edge of the seminiferous tubule. They have a nice dark nucleus. And other than that, they're just kind of a boring cell that's sitting on the edge. Nothing too distinct or distinguishing about it, so its location is going to help you a lot. It'll always be on the edge of the seminiferous tubule. That's cell number 1. Cell number 2 is the primary spermatocyte. So once the cell, the spermatogonium, is stimulated to go through meiosis, it turns into a primary spermatocyte. And primary spermatocytes are large. They're the biggest cells in the seminiferous tubules. And they have condensed chromosomes that are really visible under a microscope. So remember that DNA in most cells, if that's the nucleus and this is the cell membrane, DNA is usually a jumbled mess. It's not all packed in these X-shaped chromosomes that you see in pictures, but when you go through meiosis, they have to condense. And so you'll see these chromosomes all mixed in here, and it will appear kind of like a jumbled mess. There'll be light patches and dark patches, almost like this granular appearance in the nucleus. And what you're seeing there, it's almost like tarantula legs is what it looks like to me, like a bunch of tarantula legs shoved inside of a bowl. And so there's going to be this spotty appearance that looks very different than the spermatogonia. And so that can help you know when you're looking at a primary spermatocyte. It's the biggest cell, and it has condensed chromosomes that are pretty easily visible. So that's cell number 2. Cell number 3 is the secondary spermatocyte. It's basically just a mini primary spermatocyte. Basically looks the same, but it's smaller. It's further, or I guess deeper, into this seminiferous tubule. It's smaller because this one big primary actually splits into the 2 secondary spermatocytes. So they're almost always much smaller. The importance of the secondary spermatocytes is that this is the first cell that is haploid. Primary spermatocyte hasn't gone through that division yet. It hasn't gone through meiosis 1 yet. Once it does, it turns into a secondary spermatocyte that all of a sudden is haploid, and therefore you got to protect from this point on. So down here is where autoimmune